Welcome back, learners, as we continue the revision of KCSE 2016. Now, we stopped at question 10, and now we're going to question 11, where we're talking about name two benefits that a parasite benefits from a host. The relationship between a parasite and a host is called parasitic modes of nutrition. We have different types of modes of nutrition. There is parasitic mode of nutrition, there is saprophytic mode of nutrition, there is holozoic mode of nutrition. So the relationship between a parasite and a host is called saprophytic mode of nutrition. And there are two things that a parasite can benefit from a host. So if you're watching our channel for the first time, kindly take time, subscribe, like and share. You can also share with your friends from other schools. And I hope you're going to enjoy your lessons. So just as I was saying, there are two things that a parasite can benefit from a host. Remember, a parasite is like a tick and a host is the cow. So the relationship between a tick and a cow is called parasitic mode of nutrition. So there are two things that a parasite can benefit from a host. Number one is food. Food, which you can also call nutrients. And the second thing that a parasite can benefit from a host is shelter. So like a tick, it obtains both food and nutrients from the host. Not like the symbiosis mode of nutrition. In symbiosis mode of nutrition, both the two organisms are benefiting. But in parasitic mode of nutrition, it is only the parasite that benefits, but the host gets nothing. Okay? Now you go to question 12, you are asked, other than quadrant method, give two methods that can be used to estimate population of grass. This question was very tricky. If you are asked of grass, now if you write capture-recapture method here, you will not get a correct answer, because capture-recapture method is used to capture mobile or moving organisms. Grass do not move. So if we are estimating the population of grass, and remember the question has said other than quadrant method. Remember we usually have four types of methods of estimation of population. That is capture-recapture method, quadrant method, belt transect method, and line transect method. Because I've ruled out capture-recapture method, and already quadrant method has also been ruled out by the question, because quadrant method can be used to estimate population of grass. But the question has said other than. That means quadrant method should not be the answer. So the other two answers will remain as line transect method and belt transect method. Belt transect method. So that I hope has been understood very well. And then we go to question 13, where you are asked, state two factors that affects enzymatic reaction. State two factors that affects enzymatic reaction. Remember, enzymatic reactions are affected by several factors. I'm going to start by noting those factors. The first factor that affects enzymatic reaction is temperature. The second factor that affects enzymatic reaction is pH. The third factor that affects enzymatic reaction is substrate concentration. The fourth factor that can affect enzymatic reaction is cofactors and coenzymes. Cofactors and coenzymes. Cofactors and coenzymes. So those are some of the factors that can be used to affect the rate of enzymatic reaction. But you can also talk about chemical inhibitors. There's also chemical inhibitors. Okay. Those ones can also affect the rate of enzymatic reaction. And then we come to question, you see this question 12, 13a and 13b is a tied question. They are related. So the answer that you give in question 13a determines your answer in question 13b. So you are asked here, explain how one of the factors stated above affects enzymatic reaction. So you are told to only explain one factor. And that factor that you're talking about, you must have answered it in 13A. Like, you will only talk about temperature down here when temperature is in 13A. You cannot talk about temperature in 13B when it is meeting in 13A. That means you'll automatically get a wrong answer. Okay? Now, let me start with the temperature. How does temperature affect the rate of enzymatic reaction? Remember, temperature is divided into three parts. There is low temperature, there is high temperature, and then there is above optimum. So temperature will affect the rate of enzymatic reaction in this manner that I'm going to note here. Kindly note this. You'll say an increase in temperature increases the rate of enzymatic reaction until optimum. Then you note above optimum, the rate of enzymatic reaction, decreases because the enzymes are denatured. But you can also say low temperatures inactivates enzymes. I want to note that in small letters. So we'll say low temperatures inactivates enzymes, lowering the rate of enzymatic reaction. So that is very important. This is how temperature will affect the rate of enzymatic reaction. An increase in temperature increases the rate of enzymatic reaction until optimum. The word optimum must be there. Then you say above optimum, the rate of enzymatic reaction decreases because the enzymes are denatured. The word denatured must appear. And then low temperatures inactivates enzymes, lowering the rate of enzymatic reaction. When we talk about pH, you can only say that enzymes works best at optimum pH. And then you say extreme pH denatures enzymes. Okay? When we talk about substrate concentration, you can say the rate of enzymatic reaction increases with increase in substrate concentration. In short, the higher the substrate concentration, the higher the rate of reaction. When the substrate concentration decreases, the rate of the reaction also decreases. When we talk about cofactors and coenzymes, the function of cofactors and coenzymes is basically to complement the enzymes. So it increases the rate of enzymatic reaction. But when we talk about chemical inhibitors, remember chemical inhibitors, they inhibit the rate of enzymatic reaction because inhibitors compete with enzymes for active sites. So they are competing with enzymes together for the active sites. Hence, they lower the rate of enzymatic reaction. I hope you have enjoyed our lesson up to there. And if you're watching our channel for the first time, kindly take your time, like, share, subscribe. I'm also going to do another successive video on this, and I hope you are enjoying our lessons. Thank you very much. May you be blessed, and you can enjoy the rest of your day. Thank you.